Good morning mister.
Hi, good morning, Nasser. How are you doing?
هلا والله.
Good
كيف
morning.
الحال؟ صحة.
Thank you. Good morning, dear students.
Good morning.
Abdullah
Good
Al-Eid.
morning. ترى أنا أول واحد دخلت أستاذ.
I see you. I see you. Abdullah Al-Eid, Nasser Al-Shinif. Nasser Bari Al-Shinif. Musa Haji. Musa. Hussein Al-Qahtani. Welcome back, Hussein. Hussein, I see that he is discussing this conversation with me from the bed.
What conversation?
Yeah. Just you look very sleepy.
No, I'm awake, totally.
Yeah, Khalid Satam. Good morning, Khalid Satam.
Good morning, Mr.
Okay, I may start my session now, or should I wait for 1 minute or 2
No,
minutes?
wait mister for 1 to 2 minutes.
Okay.
Okay.
ايش فيك زعلان طيب؟
فيك عصبت شوي.
خلاص.
Okay. Khalid Al-Harbi didn't come yet. Khalid Al-Harbi just arrived. Yes, Khalid Al-Harbi. Good morning, Khalid. Khalid Ahmed Al-Harbi arrived. Abdulaziz Al-Muhayya. Good morning, Azuz. I need 1 or 2 more to start my session. Khalid Habib didn't arrive yet. Al-Walid. Good morning, Al-Walid.
Good morning.
Okay. Wonderful. Okay, I have to start now. This will be enough. So the rest will come to join us. Bismillah ar-Rahman ar-Rahim. وصلاة وسلاما على سيدنا محمد النبي الهادي الأمين صلى الله عليه وسلم وعلى آله وصحبه أجمعين. عبد الرحمن. Good morning, عبد الرحمن.
Good morning.
Yes, عبد الرحمن. Today, dear students, I'm just trying to revise fast. Hello, Ayman Nawaf. Welcome back, Ayman. Today, I'm going to make a fast revision of what we discussed, then come to continue with what we are going on to discuss. Like usually, our essential questions of the previous sessions was what are the different tools and processes used in genetic engineering? How does genetic engineering manipulate recombinant DNA? What are the similarities between selective breeding and genetic engineering?
السلام عليكم.
عليكم السلام.
عليكم السلام ورحمة الله.
Who is there?
وعليكم السلام.
Khalid Habib.
Yes.
Yes, Khalid Habib.
Yes.
Khalid Habib Al-Anazi. Yes.
Yes.
Okay.
مستر سجلتني؟
عبد الرحمن راشد. Yes.
Thank you.
مستر سجلت.
Rakan.
Yes.
Khalid Al-Bayz.
Yes.
Yes. Welcome back, Rakan. Actually, last session we discussed really, it was amazing to speak about the breeding, the inbreeding, and I think that session, I didn't want to be involved in it because the questions, what you asked exactly show me that you understood the session. So now we are going on to come in the genetic engineering, how we can get benefits of this study, this field, this way of life. It should be a way of life, a way of science. It is really something really unbelievable. So we are going on to discuss these 3 or 4 points. What are the different tools and processes used in genetic engineering? How does genetic engineering manipulate recombinant DNA? What are the similarities between selective breeding and genetic engineering? How can genetic engineering and biotechnology be used to improve human life? This is the most important one. The last question is the most important one. Let's come to see some new words here. What is meant by genetic engineering? What is the genome? What is the human genome exactly? What is the restriction enzymes? What is gel electrophoresis? What is recombinant DNA? What is plasmid? What is DNA ligase? Transformation, cloning, polymerase chain reaction, and transgenic organism. The genetic engineering. Genetic engineering is technology, one of the technology that involves manipulating the DNA of one organism in order to insert the DNA of another organism. Again, genetic engineering is a technology that involves manipulating the DNA of one organism in order to insert the DNA of another organism. Hassan Qahtani, what do you think? What can you understand from this, please?
I understood that we can like fix specific genes in humans and change genes.
Maybe you would like to say that what does mean manipulate? Manipulate means correct, yes. Manipulate here means correct the DNA of one organism in order to insert the DNA of another organism. Did you understand it now? Al-Walid, did you get the idea?
Yes.
Wonderful. Okay. The inserted DNA of another organism is known as exogenous DNA. Exo means something from is outside. Genus is something related to the genes. So exogenous DNA. And this exactly we'll discuss it later, step by step. Genetically engineered organisms are used to study the expression of a particular gene. Very important to know this. This question is really very important. The question could be coming to you here. This is really important. Really important. So I may ask a question like this. What are the benefits of genetic engineering? So your answer should be like this. This is the question, and this is the answer. Genetically engineered organisms are used for study the expression of a particular gene, investigate cellular processes, investigate the cellular processes. For example, there was one kind of organelles inside the cell, and nobody knows its function. Even GC, we discussed DNA, and we discussed the regulation of DNA, and we discussed how the protein is synthesized. We discussed it in the previous chapter, but no one of us thought what is Golgi apparatus function. All of us were concentrated only about the ribosomes, the factory of producing protein. But no one of us thought about Golgi apparatus. Okay, Golgi apparatus. Yeah, what is Golgi apparatus? Where is it located in the cell? What is it? What its function? Okay, something really serious. Who can help us to know that? Only genetic engineering. Okay, investigate cellular process, study the development of a certain disease. Yes, this is true. Select traits that might be beneficial to humans. I think you see in this scientific movies, what we have in the Hollywood movies, some of them, they are discussing, I think you watch one of these films which is called Max 1, Max 2, Max 3, this one, and Matrix, even Matrix part 1, 2, and 3. It's showing you that we can do and undo. We can do in humans and undo. So we can add traits to the humans to be something really looking like extraordinary. You understand? Something extraordinary. Okay, that's what I want you to know. The DNA tools. Genetic engineering can be used to increase or decrease the expression of specific gene in selected organisms. Again, genetic engineering can be used to increase or decrease the expression of specific genes in selected organisms. An organism's genome is the total DNA and the nucleus of each cell. The total amount of DNA in the nucleus of each cell. Khalid Al-Harbi, are you here?
Yes, Mister.
Okay. Is it clear for you what is meant by genome?
An organism is total
Is the
DNA
amount
in the
of
nucleus of
DNA
each.
in each nucleus, in each cell. Okay?
Yeah, it's clear, Mister.
Yeah. DNA tools can be used to yes, Laid?
It's not clear for me, genome. I don't know what's genome.
Genome, you know, if you look at the human genome is 46 chromosomes inside the nucleus of the cell.
It's the amount of DNA.
The amount
in
of
the
DNA,
nucleus.
the total DNA in the nucleus, you understand, of each cell,
How can you count the DNA inside the nucleus?
You can by counting the chromosomes. For example, humans,
Okay.
we have 23 pairs, means 46 chromosomes. You understand now?
Oh, yes, yes.
You
Thank
got the idea?
Yes.
Just step by step, you will know everything. Just step by step. In this session, you will know everything. DNA tools can be used to manipulate DNA. Manipulate means correct. This means that there could be something wrong in the DNA. Yes. What is making what? What's making mutation? The mutation can be good or can be not good. You understand? Something can be showing that this person is a very special person, or he is looking like disabled. You understand? So DNA tools can be used to manipulate DNA and to isolate genes from the rest of the genome. Yes, it is easy to isolate one gene. You got the idea now? This is exactly as much as it has benefits, as much as it has harms, because you know, a part of DNA can be. I see somebody is working here. Just a moment, I forgot to do it. Okay. Yeah, now it's okay. Look, for example, about this pandemic disease, what we have in the world now, this COVID-19, coronavirus 2019. This virus, for example, they are speaking in the media, and they are telling that China create this virus. Then the American, they are saying America, they are the reason for that. They made it like a biological war to destroy the economy of China, and too much bargaining, too much blah blah blah. Something exactly is not touching us. But if you look at the field, what we are now, this this is possible. During this DNA, during this, when they are checking about this gene to isolate one gene or some, this gene is a sequence of RNA, just a sequence, a short thread, a short strand of DNA. But maybe this thymine is replaced with uracil, so it became RNA. When it is becoming RNA, can be separate, moving away, then multiplying or making a replication itself and making a virus. So something created or synthesized from nothing, from this.
So, Mr.
Yes.
You're saying that the virus is not from the bat?
Yes, I said that this virus is found in the cats, it's found in dogs, it's found in bats, it's found in rats. It's not only bats, but I'm just discussing with you that it is easy to create it or to synthesize it inside this labs. You understand? It's easy to synthesize it, as I told you. You see, just this, it was cut in, it's coming unconsciously. It's coming they didn't want this, but this is happening out of their control. You understand? They are trying to manipulate the DNA. There is something wrong with this DNA. They want to manipulate it. So when they are going on to manipulate it, okay, a part of these genes are leaving. So where is it? We don't know. Okay, you have to synthesize you have to, oh my God, it's going to the air, move to the so it's spreading like this. You understand?
Yes.
Mister, they say that America are studying a new
what?
medicine for the corona, and they tried it in a mouse, and there is advantages.
This is long ago. Not only America. America is doing that. Germany is doing that. France are doing that. China is doing that. Many countries are doing that in reality. But they didn't even try it on humans till now.
Yeah, they tried it on
monkeys. It's taken monkeys. Okay. Also, the problem that this COVID-19, it is making a mutation so often. You know that the world now, they have 8 copies of it right now. And just yesterday, they discovered there is the 9th one. The 9th one appeared yesterday only. The 9th copy of this coronavirus, that it can spread through the air. Can you imagine?
Through the air?
Through the air. It's something really serious. So
it is very serious.
Yeah, it's very serious and very dangerous. It is really pandemic. It is pandemic. You know what does mean pandemic? Wabah. Okay. Restriction enzymes. Let's come to discuss the restriction enzymes. Restriction enzymes are proteins that recognize and bind. So first they are recognizing, then binding to specific DNA sequences and cleave the DNA within that sequence. So it is like that, for example, they are trying, for example, they have one kind of sheep. This kind of sheep, Australian sheep, for example. This Australian sheep is huge and it has too much meat, really, more than fats. It has too much, please, I see that there are many of you are sending some questions here. Hussein, who are you?
Mister, there is
some guy who named Muhammad.
Who is that Muhammad?
Who's Muhammad? I don't know.
We don't know.
Is he here? Just
Yes.
Yes.
I want to see him. Muhammad. Yes. Who is that Muhammad? Hello? Muhammad.
If you can kick him, it will be good.
Exactly. Remove him immediately.
Yes.
I'll go.
طالع الخواف.
Doesn't matter. It doesn't matter. You know,
Fine.
too much corruption. This is normal. Okay. So restriction enzymes are proteins that recognize and bind to specific DNA sequences and cleave the DNA within that sequence. I just want to tell you something. For example, we know that Australian sheep, they are huge enough. They have, even in Canada also, they have much meat, I mean much muscles than fats. So it could be nice for us to increase the amount of it. It would be nice and in the feasts what we have and something like this. So they decided to make they decided to go to check about its DNA, then check which gene is increasing the amount of muscles like this as a kind of as a meat. Then they are modifying the DNA of the new generations of the domestic sheep we have here in Saudi Arabia, for example. You got the idea? So by doing that, they are doing it according to what or by what? What are the tools? The tools are restriction enzymes. So this restriction enzymes are proteins that recognize and bind to specific DNA sequences. So it is just join a specific DNA sequences and cleave the DNA within that sequence. You got the idea now, dear students? I hope so. They are used as a defense mechanism by bacteria against viruses. Oh my God. So now I think we know it nowadays. We should know what is going on nowadays. Scientists use restriction enzymes as powerful tools for isolating specific genes or regions of the genome. That is true. Look here in this picture. I think, for example, this is EcoRI means Escherichia coli restriction enzymes. EcoRI is a restriction enzyme that specifically cuts DNA containing the sequence of GAATTC. I think you know what that means, GAATCC. It's clear, yeah? Sticky ends or single-stranded DNA sequences are at the end of fragments. Look at it here. It's looking really clear. So it cut it from here. Okay? Can be reattached to complementary strands. Okay? Just a moment. Yeah. The gel electrophoresis. Gel electrophoresis.
Mr.
Yes, Khalid.
I don't get the last point.
Huh. Let's come back
Yeah,
to it.
this one.
Yeah. I told you this is a kind of restriction enzyme that specifically cuts DNA. So its function is to cut the DNA. Look at it here. It is cut already here. They said this a strand of DNA. To cut it according to what it's responsible for what? For GAATTC. Guanine, adenine, adenine, thymine, thymine, cytosine. How we know that this is DNA or RNA? If there is a thymine, this means that this is DNA. If there is uracil, this means that it's RNA.
RNA.
Yeah. So it is, look at it here, GAATTCC. So it cuts it off, GAATTCC.
Ashraf.
You understand? So it is, you know, cut it. Why it is cutting it? To attach it with some new genes, okay? What do you want to add? You got the idea now?
Yeah.
Yeah.
Thank you.
You're welcome. An electric current is used to separate DNA fragments. according to the size of the fragments in a process. This process called gel electrophoresis. So what is gel electrophoresis, dear students? Can you please repeat this, Abdullah Al-Eid?
Gel electrophoresis is an electric current used to separate DNA fragments according to the size of fragments.
Wonderful, wonderful. I think it's clear for you all now. When an electric current is applied, the DNA fragment move toward the positive end of the gel. This meaning that the gel, it has a positive ends and negative ends? Yes. You should study this in chemistry. I think, inshallah, it should be this year, but I think Mr. Ahmed will continue with you about it next year, inshallah, in organic chemistry. Because I'll ask him to teach you some of this organic chemistry, because without organic chemistry, you will face a big problems in the future if you want to be specialized in this scientific fields. The smaller fragments move further faster than the larger ones. This is true. The unique pattern created based on the size of the DNA fragment can be compared to known DNA fragments for identification. Okay, this is just telling you the mechanism of it, how it is happening.
Mr. Ashraf,
Yes.
there is some guy joined. He's called Charbel B.
Charbel B, yes, mister.
Again?
Maybe he's the same guy, but come in another name.
عاد مو فاضي نزيد تطبيق من ذا اللي كل شوي يتهكر.
He escaped now.
No, he's here.
لا واحد
كيف؟
لأن عنده
ناصر خليك جيد وحرش بينهم.
ركز بس اقول.
لو ان الرجال شغل كاميرا، يلا شغل كاميرا.
مين؟
لو مستر
يلا ما
هذا.
Okay. I exactly removed him now. Okay?
يطلع مشرف يا عيال.
Doesn't matter. He should identify himself, whatever he is.
تخيل.
Hey guys, whatever he is, he has to identify himself.
I
Okay.
agree.
Thank you guys. If you see anyone, any corruptor, okay, just let me know. When an electric current is applied on DNA, I think this exactly explains it well. Okay, let's move to the next page. Recombinant DNA technology. DNA fragments from different sources can be combined to make new DNA molecules. Okay. The newly generated DNA molecule with DNA from different sources is called the recombinant DNA. So what is the recombinant DNA, Musa Haji? Master Musa?
Yes, mister.
What is meant by recombinant DNA?
ما علق عندي أنا بس صح؟
إيه ما علق عندي.
ايه
Hey guys, Musa, now I think the sound is clear now. What is meant by recombinant DNA?
It is the newly created DNA from different kinds of DNA.
Wonderful. So it can be collection of many kinds of DNA, and they are mixed together to make a new DNA. This is called recombinant DNA. Wonderful. Okay. Let's come here to watch this together. Recombinant DNA technology. Recombinant DNA is placed into bacterial cells for study via a carrier. Common carrier include viruses and plasmids. And what? Plasmids. We will know what this means, plasmids now. Small, circular, double strands DNA molecules that occurs naturally in bacteria. And yes, this is called what? Plasmids. DNA ligase. What's DNA ligase? I think from this is exactly wonderful.
Enzymes.
Excellent, Hussein. Excellent. So DNA ligase, a cellular repair enzyme. A cellular repair enzyme. So this is enzyme for repairing the cell or repairing the DNA. Attach the recombinant DNA to the plasmid. Look at this. This is plasmid. Look at it. Do you see it? This is a plasmid. It's opened, then combined with a new strand, then closed one more time. So what is connected it again? This is exactly, or repairing it. This is exactly DNA ligase. I think this is looking clear. Am I right?
Yeah, it's more than clear.
Wonderful. Should we stop here then since and continue next, or would you like to continue for some more times?
Yes,
I think this is enough.
Yeah.
You need to digest.
Yes.
You
Yes,
need to
yes.
digest this, yeah.
Yes,
بس أشرف
Okay, so we stopped with page 10.
احسن انا اجاوب على سؤالك عادي بعدين.
فهد العواجي, you just arrived. Okay.
أنا
Okay, Fahd.
من زمان والله جاي.
ما فاهم الدنيا اللي فاضل.
والله العظيم من زمان لا تكذب.
كذاب.
تبي تتكلم يا زين.
كذاب.
Okay,
طيب
guys.
والله اني جاي من زمان.
نصاب كلام
والله اني جاي من زمان بس
الحين صح.
والله من زمان.
See you then, guys. Bye.